This is an exam question about plant transport. It says figure 1.1 shows the outline structure of some cells from the phloem of a dicotyledonous plant. And then we've got figure 4.1 and we've got a diagram that clearly shows two different types of cells. The first part of the question says label the following structures on the diagram using label lines, and we've got sieve tube, sieve plate, sieve pore, companion cell, plasmodesmata. So they're the components that we've got to identify on this diagram. So plasmodesmata are the gaps between the cell walls of cells. So you can clearly see the cell walls, and we've got the gaps here. So the gap there will be plasmodesmata. So that's where part of a cell where there's no cell wall or cell membrane, but the cytoplasm goes through both cells. We've then got companion cell. So phloem is made up of two types of cells, a sieve tube and the companion cell. And this cell here is the companion cell. Companion cell is just a normal plant cell. So this part here is the companion cell. We've then got to label a sieve pore. Sieve pores are gaps. So we've got the sieve plates here. The gaps between the sieve plates are the sieve pores. So we need a label arrow. And that's the sieve pore. Sieve plate is what at the ends of the cells are broken down, so you've got gaps between them. So these are the sieve plates. Again, just a label line. Label it sieve plate. And finally, the sieve tube. Well, we've got the companion cell. So this cell here is the sieve tube. Okay, so we've labelled all 5 parts, and the question's worth 5 marks. Second part of the question says, complete the drawing by adding cytoplasm and a nucleus or nuclei where appropriate. So we've got to add cytoplasm and nucleus, and then we've got to label these components. Well, as we said before, the companion cell is a normal plant cell, so this will have a nucleus. So we'll draw a nucleus, and it asks us to label it, so we'll label that nucleus. Inside the cytoplasm, inside the companion cell is cytoplasm, so we can label this cytoplasm. And then the final thing we'll add is the cytoplasm in the sieve tubes is very close to the edge, and there's no other organelles in a sieve tube. The companion cell controls, and the nucleus of the companion cell controls the sieve, all the processes that are going on in the sieve tube. So there's lack of organelles in a sieve tube. Cytoplasm is towards the edge, and that reduces the resistance of flow of sucrose. So just around the edge is the cytoplasm. So that's all the parts drawn and labelled for phloem, which is made up of companion cells and sieve tubes.